The next topic we are looking at is nutrition, including food tests. And what is nutrition? Nutrition is just feeding, and there are different modes of feeding by different organisms. As I said earlier on, nutrition simply means feeding, and there are different modes of feeding. The first one is autotrophic nutrition. Autotrophic nutrition means feeding on self-made food. Autotrophic, self-made nutrition. And it involves A, photosynthetic nutrition, in which the organism uses solar energy in making the food. And B, chemosynthetic nutrition. The organism here makes use of chemical energy in synthesizing the food. Now, green plants fall into the first group, A, because they are the ones using chlorophyll to extract solar energy, and in the process, be able to carry out photosynthesis and make food. And B, the popular organisms here are the bacteria involved in nitrogen cycle, like nitrogen-fixing bacteria, such as Azotobacter, Clostridiums, and so on. Then the other group type of feeding is heterotrophic nutrition. In heterotrophic nutrition, the organism is feeding on already made food. So they are the type of feeding that the organism cannot make the food, but feed on already made food. And we have under this A, holozoic nutrition, feeding on solid food that requires digestion. And that is where we have herbivores feeding on plants, material, carnivores feeding on flesh, and omnivores feeding on both flesh and plants. Under heterotrophic nutrition, we also have parasitic nutrition. Here, an organism is feeding on its host. It lives inside or on the host and feeding from the host. The organism that is a parasite is feeding parasitically. Then we also have symbiotic nutrition. Here, two organisms live together and help each other to get food. Example is nitrogen-fixing bacteria in the root nodules of legume. The bacteria helps the legume to fix atmospheric nitrogen to make protein, and when it dies, it decomposes to produce nitrate for the plant, which the plant uses to make its own protein. We also have, in the process, the legume itself photosynthesize and give the bacteria sugar as food. That is a single example. We have other examples like lichen, bacteria in the large intestine of ruminants, and so on. Now let's look at the D type, saprophytic nutrition. Here, the organism is feeding on dead body. Fungi fall into this group. Some bacteria are also in this group. They digest the dead body and absorb the food later on. This such digestion, when the organism have to deposit the enzyme or the dead body outside the organism, is extracellular digestion. Extracellular digestion. Cellular digestion. Digestion outside the body. So saprophytes, fungi, and some bacteria. In ecology, we refer to them as decomposers. Let's move on to define photosynthesis, because from here from photosynthesis we have photosynthetic nutrition. Now, photosynthesis, as we know it to be, is a process of manufacturing food by green plants using water and carbon dioxide as raw material in the presence of sunlight. Photosynthesis is divided into two stages. Light stage, where photolysis occurs, water being split to hydrogen ion and hydroxy ion. From there, ATP is produced, reduced compound NADPH is also produced. Then the second stage is dark reaction. This is which start with carbon fixation to form 6 carbon molecules, and in the process, food is produced, mainly carbohydrates and mineral nutrients from the soil added in the process to produce other classes of food, which are carbohydrates, proteins, lipids, vitamins, salts, and water. This is part of classes of food, but the food manufactured by a plant are carbohydrates, proteins, lipids, and vitamins. Other area of interest is food test. How do we know that a particular nutrient is carbohydrate? What type of carbohydrate? Or is protein? Or is fat or oil? So we have series of reagents for testing for this. Even water, the presence of water can be tested for. In chemistry, you do quantitative analysis where we talk about different types of salt radicals, and you can also detect which salt, whether it's chloride or it is sulfate or nitrate. It can be detected in the laboratory. But look at the common ones. The food item like complex sugar, like starch and glucose, you make use of iodine solution. You turn iodine solution on them. What you have is blue-black color, which shows the presence of starch, or the substance contains starch or cellulose. The other carbohydrate that can be tested is reducing sugar. Reducing sugar are monosaccharides like glucose or fructose. This one we use reagent like Benedict solution or Fehlin solutions. When you add this solution to any substance that contains reducing sugar and you warm it, the heat, you have this coloration, brick red color. For protein, there are two popular tests. Burette test, which is addition of sodium hydroxide and 1% copper 2 tetraoxosulfate solution into the food item. Then you see the food, the mixture changing to violet color. Violet color. Another test is using Millon's reagent, which is a chemical. When you add it to any substance containing food, the first thing you see, it will congeal and turn whitish. By the time you warm it, it will turn brick red. So we have others like xantophytic test, where you use ammonium solution to test for it, an ammonium acid. Then we have another type of food, the lipid, the fat and oil. We have two tests too, but the one popular one is mixing the food item with Sudan 3 solution and heat. You have bright red coloration. The other one is using osmic acid. When you warm any lipid food substance with osmic acid, it turns black, carbon black, shows that the food contains fat. So we are now going to look at questions based on food nutrition and food test. The first question here is: a mixture of mercurous and mercuric nitrates is added to a food substance. A white precipitate is formed, which on gentle heating turns red. The food substance is A. protein, B. oil, C. carbohydrates, D. Vitamin A. Now, this mixture, mercurous and mercuric nitrate, is Millon's reagent. And Millon's reagent is used for testing protein. So answer here is A. Option A answers the question. Number 2. In a burette test, some proteins was mixed with sodium hydroxide solution. Which of the following chemicals will be added to the mixture to obtain positive results? Let's go over the question again. In a burette test, some proteins was mixed with sodium hydroxide solution. Which of the following chemicals will be added to the mixture to obtain positive results? A. Mercurous nitrate. B. Copper 2 sulfate. C. Ammonium hydroxide. And D. Sodium carbonate. Now, in burette test, 2 chemicals were mixed with the food. One of them is sodium hydroxide. The second one is not mercurous nitrate, and it is not ammonium hydroxide, and it's not sodium carbonate. So the second reagent to be added to sodium hydroxide is copper 2 sulfate. So that completes the burette test. And the food you test with burette test is protein, and it will give you valid coloration. Number 3. In addition to high calories derived from fats and oil, they are also used in producing new cells. That's A. B. Necessary for enzyme formation. C. Used in as isolators. And D. Required for growth. Now, we are talking about fats and oil. A option, producing of new cells, we make use of protein. Production of enzymes, you use protein. Growth requires protein. So, obviously, the other use of fats and oil, apart from giving us high quality energy, is used as insulator. Insulates nerves, myelinated nerves. It's also deposited in the skin to insulate the body against heat, especially during cold weather. The answer is C, used as insulator. Number 3. Number 4. An extract from food substances reacts with Sudan 3 solution to produce scarlet red. The extract contains A. Fats, B. Protein, C. Reducing sugar, D. Carbohydrate. Carbohydrates out of it because it's general term. Reducing sugar, we use a Benedict solution or Ferlin solution. Here we are talking of Sudan 3 solution. Protein, we are either using burette test or Millon's reagent. So answer is fat. Sudan 3 solution is used to test for fat. A is the answer. Number 5. The 3 important organs located near the stomach are, this question is asking about the organs of the body. Now, if you have dissected an animal, you will be able to locate the position of these organs. A. We are talking about organs in close association with the stomach. We have kidney, liver, and gallbladder for A. B. Liver, kidney, and spleen. C. Pancreas, liver, and kidney. D. Liver, pancreas, and spleen. Now, the stomach, around the stomach, kidney is far from the stomach. It's at the lower abdominal tract, near the urinary bladder. So kidney is out of it. Now, gallbladder is inside the liver. B. Kidney is also mentioned, so B is out of it. C. Pancreas by the duodenum, liver, and kidney. Again, kidney is mentioned for C, kidney is out of it. So D, liver, pancreas, and spleen. Answer is D.